So I will speak about tensor methods for omics data analysis. First, I want to kind of give them insight into why we in these days come up with very complicated, like fancy methods enters the bioinformatics domain. And because now we are in the area of biotech, right? So here in the Broad Institute also there are groups and centers. Actually, they are creating a really valuable data set and they are sharing with public. Now we are able to generate or access terabytes of data, and it's suppressed the point that we cannot just look at and try to get insights out of our data. So we need principal models to get these complex patterns in our biological data sets. So just to give them more insights into, I think audience here are very familiar with this stuff, insights into how and why now we are able to access that much data set faster than before. Think about the Human Genome Project. To get the first human reference DNA, right? It took like around 32 years. But if you look at complete 85% completion, for sure, it's way earlier, right? And it costs like millions of dollars. Every resource reported different numbers. So I prefer not to exactly say. But now, thanks to the reference we have, and thanks to the new technologies we have, sometimes it takes as short as 1 day, and depends on the technologies you use, right? It doesn't cost, in this case, that much. Now we are able to sequence, and we are able to get the data faster, but it's not just the quantity of the data is increasing. Dimensionality and variety of the data is also increasing. Okay. So here, I will give a beloved example of the Broad Institute. Mostly they use this smoothie, fruit bowl, and fruit tart example a lot. First time I think I heard from Aviv Regev, she was giving this example, and I started understanding difference between different technologies like that. So here, so we kind of started from bulk, like for sure there are different technologies, but one of the starting point is like starting from bulk genomics. When you have a tissue, we are looking at average gene expression in this tissue. They call this like a smoothie. You know what's happening on average, but you don't know, you don't have the heterogeneity. And then when you move to single cell, right now, when you move to single cell, in this case, then now we have more expression for every cell. In this case, we know like more information about heterogeneity in this tissue, right? In this case, it's like, you know that you have berries, right? You have like your macrophages, you have T cells, but you don't know the exact location. But now some technologies was also developed here. Now we have different spatial transcriptomics technologies with different resolution. They call it like a fruit tart, right? You know which kind of cells you have, and now we know the locations of these cells, more or less. Again, it depends on the resolution. And it's not just this. I cannot use this laser. I want people online also try to see the laser. Anyways, to see about it. We'll fix for the next talk. And we also have, is like now we can collect multi-sample data sets for example, different patients group. We are looking for different diseases, and we can use different omic platforms, get data on mutation, copy number, gene expression, DNA methylation. And actually, we get this multi-omics data, so you have a bunch of matrices to analyze, right? Now, it's actually, if we go more like a data perspective, we start from a vector, right? You start from a vector here for a bulk, then we are going for matrix for every tissue. Then we start collecting omics data from different platforms. You have a bunch of matrices to analyze. So the dimensionality keeps increasing. So for these high dimensional data sets, I will introduce the tensors. So we will come back to these examples and how we will discuss how the tensor methods could be useful. Okay, so what's the tensor? Again, we said we will use tensor to represent and analyze multimodal high-dimensional data sets. And tensors are multidimensional array, right? You start from, for example, scalars, like scalars, like time points. And we use most of time vectors, what we said for the bulk data, time series data, we had tag vectors, we went to the matrices. So we keep going actually, we don't stop. And the moment we go, it's like you are putting more dimensions and more variations. The simplest case will be three-way tensor. And the moment you put more dimensions, it's gonna be higher order tensors. And we said they are considered generalization of matrices the higher dimensions. So here there is a notation crisis. And sometimes I also causes notation crisis. Sometimes they say, for example, for this cube order 3 tensor or three-way tensor or three-dimensional tensor, everybody use this, how to say, their own notations. I will mostly call order 3 or three-way tensor. Okay, so here the other idea, just to give more mathematical details, goes to second talk. Here it's gonna be primary, will be more generic, and we'll go through the case studies. But mathematically, what's happening? Lots of traditional data analysis methods were matrix-based, and matrix correspond to linear maps. We use linear algebra a lot. And when you go this high-dimensional data analysis methods, tensor-based, tensors are corresponding to multilinear maps, and we use multilinear algebra tools. Actually, my PhD was like in algebraic geometry and multilinear algebra. I work very theoretical, then I discover applications during my postdoc, and I am so happy about it. So it's mostly like multilinear algebra tools. Okay, so tensors, why they start getting popular in different areas of data science, they give a compact way to represent multimodal data. For example, if you think about the color images, we have height, width, and color channels. So these can be represented by a three-way tensor. And think about, like, in the videos, we can also add the time as a different variation. So if you have the videos, it's gonna be four-way tensor. And actually in the computer vision image processing, these tensor methods are heavily used. And one of the areas I use for multi-sample, multi-tissue genomics data, you are looking at how gene expression is changing across different tissues for different individuals. In this case, we are trying to look at these triple interactions, multilinear interactions, to represent these ones. In the simple case, we can use three-way tensors, and to analyze this one, we will use tensor factorizations, which I will introduce. Another possible data sets, like perturbational data sets. In this case, you can have, so we have a cell line treated with a drug, and we are trying to understand what is the effect of this drug, right? You can look at the change in gene expression. So this triple interaction between cell line and drug and genes can be represented by a three-way tensor in the simplest case. And I will give an example. We can use tensor factorization to understand these triple interactions. Okay, there is also other domains like network learning, social computing. It was heavily used in chemistry, bioinformatics. Again, it's not very surprising. In lots of areas, we start getting high-dimensional data, and these tools start being more popular because they are serving the community. Good, so I will give some simple notations. This is, okay. First, I want to solve my problem, so with the clicker. I cannot use it. Oh, yeah, people online will not be able to see this part. Okay, so let's say, so the notations I will introduce, like similar to matrix notation. Let's say X is a D-way tensor of size N1 times N2 times ND. So N1 corresponds to the first modality, N2 corresponds to the second modality. And I will denote the I1, I2, ID to entry by X, I1, I2, ID. Right, so if you have a tensor of same size, two tensors of same size, X and Y, when I say same size, it will have same size in every modality. So you can take the inner product of these tensors. The inner product will be given as we multiply entries for corresponding indices, and we sum them, right? Actually here, I'm not gonna be able to go all the details. There are lots of, like, we have matrix multiplication. There is a different way to multiply tensors with matrices, right? It has its own operations, its own way to define rank. We will little bit speak. Lots of things doesn't directly generalize the tensors. And we will define Frobenius norm because we will use it a lot. If you just use this inner product, you can induce the Frobenius norm. It's the square of the entries we are summing, and we take the square root. We'll use it a lot. Okay, So I will go ahead. There is one more, more technical part, but most technical parts goes to second talk because I want to speak about rank 1 tensor first. So when we do tensor factorization, we will try to look at rank 1 tensors in our data. Then I want to explain why we care about rank 1 tensors, because they will give a unit of an expression pattern, and I will give solid examples about it. So first, let's start from rank 1 matrix. So if you have vectors u in Rn1 and v in Rn2, so you can take the other product of these two vectors, u other product v. It's like u times v transpose in the simplest case. So it's going to be rank 1 matrix. And we do matrix factorization. Actually, we are trying to write our matrix as sum of rank 1 matrices, right? But you can have like d vectors. Let's say u1 has in Rn1, u2 is in Rn2. You can take the other product of d vectors, and it will give you a d-way tensor. So this tensor, it's simple, and the name is also simple tensor, and they also call it pure tensor. So it is called a rank 1 tensor. Why it's important? So actually, if you look at I can look at the entry t, I1, I2, ID, right? This I1 is from 1 to N1, I2 is from 1 to N2, IT is from 1 to ND. This entry will be, you go your first vector, U1, and you take the I1 tensor. You go second vector, I2, you are getting the I2 tensor. And you go your last vector, UD, and you are taking ID tensor. So it's separable signal. So it represents a separable signal, which can be expressed as the combination of independent factors from each mode. So it will give us a unit of an expression pattern. And just to give a little bit probabilistic connections, probabilistic connection is actually more details, but I want to give the simplest case for rank 1 tensor. Let's say you have d random variables, independent random variables, right? We are gonna look at their joint distribution. You can write the joint distributions of these d variables as the product of their marginal distributions, right? So let's say P, probability of x1 is I1, x2 is I2, xt is ID. It can be written as product of separate distributions. And I can represent this one with a d-way tensor, and T, I1, ID will give this joint distribution. And if you look at how we write, we separate the marginal distribution. It's other product of these distributions. Actually, rank 1 tensor represents a system where each dimension corresponds to an independent random variable, and the tensor entries represent the product of probabilities or related measure associated with each independent variable. Okay. I'm not gonna go actually higher ranks correspond to conditional independent variables, but we're not gonna go details, but in discussions, you can ask more details. Okay, now we kind of spoke a lot about it, and we were gonna go, why is xi element of integers up to ni? So we get this question, let me go. Okay, so this is a good question. It doesn't have to be element of integers up to ni. Actually, yeah, it's kind of a labeling mistake. It can be x1 is how to say, can be represented with values, let's say lowercase x1, and it can take different values, not necessarily integers from 1 to ni. Okay. It's my typo. Thank you for pointing out. Okay. Okay, so now we have a tensor. Let's see, let's say this is the modalities of genes, cell types, and samples. So we are looking how gene expression is changing across different cell types for different samples. These samples can correspond to different donor profiles as well, right? So here, okay, what are the traditional approaches? So we are more comfortable using matrix-based tools, and traditional approach is like becoming a data ninja. We have our tensor, we slice it, or we unfolded it. But knowing the slices and unfoldings are also important because it will keep appearing in the optimization part of these algorithms. So for example, here, we can slice this one along the first modality. So let's say you have 20,000 genes. Now, welcome, you have like 20,000 matrices to analyze. So I think it's a lot of work. Sometimes people, okay, they don't go in the gene level, but in this case, instead of analyzing one concrete structure, right, multilinear structure, you may end up with analyzing 20,000 matrices. Okay, for sure, maybe you can decide, okay, I'm gonna slice this across samples, second modality. So in this case, if you have, let's say 100 samples, you end up with, in this case, okay, second modality though, cell types. Sorry, if you have 10 cell types, you can end up with 10 different matrices and analyze them, factorize them, and merge all the, interpretations, you have to merge them. And again, this is like horizontal, vertical, and lateral slices actually you can do along the third modality. This was most common approach. You get they get the data and they work on those matrices, but we are not gonna do like that. We are gonna keep the integrity of the structure of the data. The other way most people do unfoldings. So let's look at this one. We have a bunch of slices for the first modality. We kind of put next to each other and form a big matrix. So it becomes genes times number of cell times number of samples. And we can do this one for the second modality, cell types times a long matrix or samples times long matrix. These are most common approaches, not the tensor approaches, matrix-based approaches. But one thing to notice, when you matricize the data, informations on the interactions, relations between different modalities is lost. So we will have more discussion on that one. Okay, for example, I will introduce tensor factorization. I want to make sure I mention about matrix factorization as a foundation. So here, most of the time, let's say we start with this big data and people do matrix factorizations. In here, let's say there are different matrix factorization based on the constraint you put in your factors, orthogonal, non-negative. Most people prefer non-negative because it's easy to interpret the results. So here, let's say you will get genes times factors. Let's say you are factorizing to rank R. This is like low rank approximation, right? And you will have cell types over each sample times factors. So here, what happens when we flatten? We are concatenating two variations into one dimensions. If it's actually higher order tensor, you will concatenate many variations into one dimension. So here, matrix factorization, what's happening mathematically, I'm writing my matrix as sum of rank 1 matrices, right? I have here lambda I, gi, like for example, this guy will correspond to g1 times ci, right, c1. And here, lambda I tells me how strong is the signal. Gi, in this case, gives the genes latent factor. Ci gives the cell types latent factor. For the tensors, we will go very deep and we will see, okay, how we can interpret these factors. For example, here, we are looking which genes are upregulated, downregulated, what are the pathways. And then when we go cell types over each sample, and we are trying to see which cell types are active in which samples. So it becomes like trying to decode these two dimensions. Okay, here, what we are gonna do, we are not gonna flatten, we are not gonna slice. Is there any way to factorize it as it is? And yeah. Yeah, sure.
In the cell type, so cell types are mostly categorical in this case, right? They're categorical data. So we have endothelial cell neurons and different types.
Yeah, like T cell, macrophage.
Yeah. So how do you represent them in as a matrix?
Like, okay, so here it's actually, it starts with how we start our matrix. So our matrix is actually, it's like a gene expression for, let's say, T cell across this sample, right? Every time we are speaking about gene expression. In cell type latent factors, right? Actually, we will discuss this one. What happens? We are looking at what are the weights for corresponding cell types. And for example, if T cell has a high weight, we will say that this gene expression program is activated in T cell, right? Maybe I should have mentioned how I set up my matrix. But in any case, we are looking at gene expression for specific cell type for specific samples. Okay. Okay, so here we are gonna look at more generalization of matrix factorization. I will introduce two common tensor decomposition, but there are multiple tensor decomposition methods, but these are the most popular. So we will discuss in the next talk technical details. They are the most popular, not because, how to say they are the best, because they were the first ones. So like Tucker decomposition is one of the most popular and first. And let's say again, we are looking at how gene expression is changing across different cell types and for different samples. So these are data. For example, Tijk will be expression of gene I at cell type G for sample K. And let's say the size is number of genes times number of cell types times number of samples. So that one can be so big. In any case, you may start with like around 20,000 genes, right? So here, first thing we will do, we will reduce the dimension. So most of them, we are not really looking at, yeah, we work with 20,000 genes, but we are trying to find meta genes or gene clusters, right? We are trying to get these parts. I can find gene expression programs. So we reduce the dimension here. Let's say next talk, we will discuss how you reduce the dimension and how you can find the ranks to reduce the dimension. We go to R1 times R2 times R3. So here, when you do matrix factorization, from one matrix, you get two matrices. Now, when we do a simple case three-way tensor decomposition, from one tensor, we are going to get three matrices, one for each modality. And we will discuss, we also have something for tensor. I will discuss it. First, I get gene factors. It is number of genes times R1. R1 is the reduced dimension. It means you have R1 latent factors, and we will look at what they correspond to. They will correspond to a gene expression pattern. And for samples, it can be, this is really important. I care about it. So it's number of samples times R3. Let's say I have 200 patient profiles, but at the end, there are two subtypes maybe driving these gene expression programs, maybe disease, non-disease, right? Or you can have like different subtypes of the cancer that we will discuss. So this one will help you to find these main, I'll say, donor variations. And actually, it will help you to class discovery with the sample factors, and cell type latent factors will help you to see in which cell types these gene expression programs was activated. So we reduce dimension there as well. For cell types, sometimes you not necessarily reduce. So there is this thing. Mostly, this R1, R2, R3 is less than number of genes, number of cells, number of samples. It was. And for genes, definitely. But for sometimes cell types, you can even go to R2 can be greater than number of cell types. Why? For example, maybe you have a T cell. It's activated differently for disease condition. And you have a T cell, which has a different program for non-disease condition. So you put T cell as one variation, but you may end up with two T cells program. And we will discuss them. And core tensor look at the correlation between different factors. We will also discuss it more mathematically. For the matrix factorization, we have the coefficients. It tells how strong is the signal. So here we will have the core tensor to say how strong signals between different factors. Okay, let's go. Here, what's happening mathematically? I have a tensor. I'm writing my tensor as sum of rank 1 tensors. Let's see here. This is Gijk gives the how strong is the signal, and GI is the genes latent factor, CJ cell types latent factor, and SK will be samples latent factor. K is samples latent factor. So this is like a rank 1 tensor expression pattern, and core tensor will tell me how strong is the signal between these factors. So here you can have R1 different genes latent factor, R2 different genes cell type latent factors, and R2 different sample latent factor, and GIK. That's why it's R1 times R2 times R3. It's like a matter of indexing. So people summarize this one and show this is core tensors times gene factors. You put all the factors next to each other, and it becomes column of a bigger matrix as we discuss, and cell types factors and sample factors. Okay. This is one of the most common, and I want to quickly introduce another one is heavily used, CP decomposition. It's a special case of this one. That's why I started from here. In CP decomposition, everything is oversimplified. It says, okay, ranks over each modality is the same. R1 is equal, R2 is equal, R3. Sometimes it works perfect. And if it doesn't work, we go to Tucker, actually. And in this case, it's like, then here you don't need the core tensor, you just need the coefficients. Here, Gijk is not equal 0 if and only if I is equal, J is equal K. So we just end up with lambda I times Gi, Ci, Si. I'm going to introduce more visually. This is called Actually, if I start discussing what is called, it would take 1 hour. So everyone, different research groups, they discover this method at different times, and they all thought that they are the first one discovering this method. There is a long list of names, but I'm gonna give the most common one: canonical decomposition, parallel factor analysis. They shorten it, they say it's CP. But if you see a paper and if you see, okay, they are calling it in a different way, it's not my fault, okay? I just say what is common. And most of the time they use alternative least square to solve these questions, which in the next talk we will discuss details. That's why they call CPLS. In CPLS, just to go quickly, I start from my three-way tensor, and I'm getting 3-factor matrices. All of them has the same reduced rank. And mathematically, actually, I'm writing my tensor as sum of rank 1 tensors. You cannot really write, because we are working on noisy data, we don't really write as equal. We are looking at approximation. Next talk, we will discuss how good is approximation, how we can choose rank, right? And we also have noise. We will discuss about, okay, which kind of assumptions we will have on the noise and on the factors, which could work for genomics data. Okay. And now, actually, in this talk, I will mostly go over some case studies. There are many applications of tensor factorization in omics data analysis, but I will try to give 4 or 5 applications. But first, I keep speaking about rank 1 tensor. Let's see what rank 1 tensor represents. So this is more detailed version of this example will be given in our talk. It is from our project, classical Hodgkin lymphoma. Let's say we have this tensor, genes times cell types times samples. We were working on over 20,000 genes for 10 cell types and 40 samples. Like this is 40 tissues taken from different patients. So here, one of the rank 1 tensor appeared in the factorization. For example, here we had, again, genes latent factor, cell types latent factor, samples latent factor. This is a unit of an expression pattern, but how does it look like? So genes latent factor has length 20,000, and we look at which genes are upregulated, downregulated, or which gene clusters together upregulated, downregulated, and you can look at the pathway. We find the pathway, which is JAK-STAT pathway, promotes tumor cell proliferation and survival of tumor cells. So this is the first goal. We find gene expression program. So actually, in one shot, you can look at all the triple interactions simultaneously. Then next question you ask, okay, this program is activated in which cell types? And we go to our cell type factors. Here, okay, so the height of corresponding to these different colors correspond to these 10 different cell types. The height actually gives what is the weight, at what is the loading. So actually, it shows that only tumor cells has high loading. So we say this gene expression program activated in tumor cells, which makes sense, tumor proliferation. Right, then we look at, okay, this is activated in which patient groups? Then you'll go look at your samples latent factor. So here again, these heights correspond to the loadings, and we have 3 groups. First group of patients, again, is unsupervised, so we are not really putting labels in our algorithm. So it is Hodgkin lymphoma patients, and dark blue ones, Hodgkin lymphoma patients with EBV virus, open blue, Hodgkin lymphoma patients without EBV virus, and orange one, reactive lymph node patients. And this actually says, okay, this program is activated in Hodgkin lymphoma patients. So even if you didn't know labels, so we know it, but we didn't use it. That kind of unsupervised method, it's already, they're able to find the stratification of your data, donor conditions. So that's how we are gonna use it. And in the applications, these patterns, this kind of rank 1 tensors will keep appearing. Let's go. I will have 3 slides to kind of compare it with a couple of some methods, but more details will come in the next talk. So again, we already discussed non-negative matrix factorization and PCA. Most of they unfold the data, and you can lose the heterogeneous, interactions between different modalities, and interpretation becomes very hard. You merge the analysis from your multiple unfoldings or multiple slices. And non-negative matrix factorization is not unique. Here, okay, I am always in the tensor domain. Also, I create some conflicts. Most of the time when you read tensor methods are unique, this is too theoretical. So they will say, okay, one of the, so I'm both speaking limitations, I'm speaking truth. They will say, okay, NMF is not unique, but tensor methods are unique. First, work with the noisy data. You will see if it's unique or not. It's unique theoretically, but it's not stable. Next talk, we will go over it. But theoretically, it's unique under milder conditions.
Stable? Yeah, in this
Stable,
case?
yeah, we will discuss it more. Stable is like when you run the same algorithm, you don't always get the same factors because it will be heavily dependent on the initial guess. Right? Theoretically, it may be unique, but it's not going to be stable, and we are going to address it. And again, rank selection is non-trivial for both of them. And for tensors, we have a pipeline to discuss. And the most important thing, higher dimension is challenging interpretation for matrix methods. And for deep learning approaches, in the deep learning approaches, there are two things. One thing, it depends, like it's evolving, but latent space interpretation sometimes is very difficult, not even possible. Here, we already discussed our latent space. Rank 1 tensor already was giving us latent factor. It's very interpretable. Other thing for deep learning approaches, sometimes you need thousands of data, right? You need lots of funding, but sometimes we work in some public data, or one of the projects we work on glioblastoma. It's not easy to get so much data, right? You can also work with small numbers of data sets with these methods, because in deep learning, inference on patterns on a new data set is as good as training data set. In the cases you don't have a big training data set, factorization-based methods could be utilized. And the last thing, differential expression methods. For the differential expression methods, for the gene expression, you have to give the groups. You say group A versus group B. It's you already know. But when you have a new data set and you don't know what is driving these gene expression programs, you don't even know the subtypes of cancers. In this case, unsupervised methods like factorization methods will be more beneficial. Actually, we will have concrete example. Here, for DE methods, you need to give predefined condition, and it doesn't consider sample heterogeneity. Maybe I have two patients, but they can also have heterogeneity between them. So it's actually kind of hurting the whole analysis. Good. So I will now give some examples of the applications, some case studies. One of the common examples goes with multi-sample gene expression data sets, and it can be, you can consider multiple cell types or multiple tissues. Here, for example, we start from a single cell data, right? You can find the cell types, and we may have, it's so similar to what we discussed, like gene expression over different cell types for different donors. Okay, so there are actually lots of sources to get this kind of data sets. GTEx, Stevens UK cohort, Illumina Body Lab, and human tissues, for sure TCGA, Cancer Genome Atlas, right? Again, as we discussed in the previous slides, traditional approaches often assume that gene expression patterns remain consistent across different contexts, right? Or samples are independent or homogeneous, but we also, in tensor factorization, we respect the heterogeneity. So if you structure this one as a matrix, you have some challenges. It may hinder to revealing cell type specific, tissue specific, or individual specific patterns. So we want to really go donor specific or condition specific patterns. Also, you can, if you do matrix size, you can ignore individual heterogeneity. In one of the, I think, next case study, they're actually looking at how these gene expression patterns are driven by biological factors like race, gender, age, right? You may need some correlations. So there are, okay, this is the main pipeline, but it's similar to what we discussed. We factorize, right? And in this case, maybe I'm not gonna go over it again. After factorization, we are trying to see the meaning of our latent factors. So here, for example, you are looking at gene factor 3, and so here I just put top 20 genes, but for sure you do like a pathway analysis, gene set enrichment analysis to find what is the function there. And you go to cell types, latent factor 3, to see, identify cell types with highest loading. You say this pattern is activated there, and you look at your donor conditions, like donor latent factor 3. Here, you can test for, you look at covariate effect, test for correlation of these donor factors with whatever clinical information available, healthy disease, like a gender, right? This kind of things. And this is a case study, multi-cluster. I wanted to also give some like studies so people are more interested in reading a lot around, so it will give a resource. So again, they had GTEx data, and they had 544 individuals across 53 human tissues, including 13 brain subregions. So it's like 18,481 times 544 times 53 tensor. And they did tensor decomposition. I guess they were, I'm not totally sure, they might have just reduced up to 10 main factors, right? They used something called semi-non-negative tensor decomposition. This is what it means. For the tissue latent factor, they force non-negativity so they can interpret the results easily. So means in my application, I force non-negativity for every factor, so the interpretation is easy. Okay, again, they did the same, find gene clusters, tissue cluster, individual clusters, and they test the correlations of the individual clusters to clinical attributes. They use tensor methods here. You can check more details for clustering, classification, covariate effects. For example, for clustering, there are multiple groups in this study. This is in A, they do gene clustering, tissue clustering with PCA. It's one of the cases because of loss of heterogeneity, as our clustering doesn't become very perfect with PCA. Also, we will discuss people also assumes normal distribution. It's not totally true for genomics data. Here, the clustering with tensor factors, they show it becomes like more, you can see your more identified clusters, but they also look at different tasks. Again, you can see details here. And I want to quickly go another application. I want to cover at least 3-4 applications. We can discuss later details. Another application I really like, multi-omics data. Now we can get data from genomics platform, transcriptomics, epigenomics, proteomics, metabolomics. I couldn't say it. Again, there are different resources to get this kind of data. But now, actually, we are like, okay, for one patient, I have more and more and more information. Okay, but can we use all this information? Right now, for the tensor methods, what we are gonna do, we will try to, there are different ways to merge though. We will try to merge this data to a big tensor, and we will try to do factorization. And yeah.
So when we get DNA methylation data, for example, it's usually not on the
not gene level.
Yeah,
like
not gene level, right?
Yeah, most of the time they do more gene-centric, right? For example, most of the studies, Amir, we are working on something like that now. We are looking at average methylation for gene level.
Okay,
yeah.
So they try to convert.
Yeah, you try to find a
gene.
common modality. Lots of time merging happens gene level. It's a good question. Thank you. Yeah. Missing points.
Thanks. A lot of these data sets have obvious links between entities like genes, proteins, methylation sites. Can this be taken into account?
Yeah, this can be, can you ask again? They are, yeah.
Yeah, so you can draw clear links between a gene and a protein, for example, and then you have measurements in these samples. Is those links something you take into account in the model?
Like, if, for example, you can also even give, this is more regularized tensor factorization, protein-protein interaction networks, or other kind of maps you have. It can also learn a map, but it is, that method is a little bit more complicated from what I discussed. But there are actually methods like that. They learn from the like graphs, right? The relation between different proteins, your genes and proteins, and they kind of like gives you more inference. Right, but thank you. Okay, here, one of the way, again, the way you merge, it also depends on your data, and I will give one example, and there are different styles of merging. Actually, now we have this kind of data set, and we are trying to merge by ourselves. It's like here, let's say we are merging these data sets. Again, you get a big tensor, but what happens when you factorize? We will discuss again. Again, this is like we form a big tensor. It's not like you have access to all of them. Like once you have access, then you are trying to find overlapping information for the, like, for example, trying to say, okay, 20, let's say 2000 genes and expressing all the profiles, and we try to find overlapping matrices, like overlapping modality. Okay, here, again, we have some donor matrix, right? And we have donor latent factors and gene matrix, gene factors, and omic factors. And here we get GR, DR, OR, and if you look at this rank 1 tensor IJK, it captures the interaction between the I gene for J donor, but K omics platform. but I will explain what it can mean and which kind of results we get from it from the case study. This is a commonly used case study, Monty. It's multi-omics non-negative tensor decomposition for gene level. They go, okay, for the methylation, you have information based on the promoter, but they go to gene level integrative analysis. So they, for example, had gene expression, methylation, miRNA, and they factorized this one. And when they factorize, actually, what they try to find and what they find. In this case, so their data has 597 breast cancer, 314 colon cancer, and 305 stomach cancer. Actually, they form 3 different tensors for them and work them separately. And most importantly, for example, for breast cancer, they are looking for 4 different subtypes, right? They have luminal A, luminal B, HER2, and basal. And Monty, in this case, was able to detect subtype specific or clinical attribute specific, right? Because we have more information about the donors we can later use, gene sets that show strongly regulated by certain omics. And it also shows, for example, it was both strongly regulated by methylation and mRNA expression. So you can see also kind of correlation between different omic types. They can refer the like same type of gene regulation. And it's kind of like main conclusion is integrating multi-omics data in a gene-centric manner improves detecting cancer subtype specific features and other clinical features. Yeah.
Is there a reason why they construct different sensors for the 3 types of cancer?
They can, yeah. So you can also put all of them together. Actually, then in this case, it's not a lot of data. It can find breast cancer subtypes, colon cancer subtypes, and stomach cancer subtypes. But maybe when you work in a smaller data set, I'm not sure if like finding subtypes becomes easier. But I think mostly our approach is kind of approach. We put all the data together and we first run. We will say, can the methods tell us, right? Okay, this is actually kind of 1 cancer group, 2nd cancer group. It's like, I think there is no need to actually make 3 cancers. But like, again, it's like a try. I would put all of them together, but at the same time, I would put them separately to make sure, am I getting more information like that? Or when you put them all together, maybe you can also put more noise in the data, right? Okay, so I have, how many minutes? I don't know.
Maybe.
Good, okay. Data imputation, and we will have cell-cell interaction. Cell-cell interaction will be the last application, but there are more applications here. Data imputation, so here, one of the projects we work, so let's say CMAP, DepMap, you have lots of like data for drug effects, right? You can have a drug, a cell line treat with a drug, and you can look at gene expression change before treatment and after treatment, right? Then in this case, we can say actually what is the effect of this drug. So you have this triple interaction drugs, genes, and cells. And it has lots of missing data because even developing a drug, right? So it takes lots of time, lots of funding, sometimes 10 years. Actually, during COVID, we were really saying, okay, drug repurposing must be really good. It's like we were really giving importance to drug repurposing. Sometimes you don't even have time to develop a new drug. So can we learn the signatures of the other drugs to infer something about like, okay, this drug, actually in the Golub lab, I think Stephen Corsello had the paper, right? He was showing some non-oncology drugs can be used for cancer as well. So this is the main biological goal, but it's a mathematically missing data problem, right? So here in this kind of, this is a paper, try this one. And in this kind of missing data problems, people try different things. These are kind of simple methods, but one of them actually, you have your tensor, can you find your low rank approximation and use this approximation latent factor to rebuild your tensor. But I will also discuss there are multiple limitations in these approaches as well. Here they just use CP decomposition. It might not be best way to approach this kind of questions. The self-specific prediction and application of drug-induced gene expression profiles. This is the paper. So I will discuss using low-rank approximation directly might not be best, because next talk we will discuss that CP decompositions are not convex. Also, let's say you have a matrix with totally empty column. when you do factorization, this empty column, when you try to input, will come as 1, 1, 1, 1, 1. Right? You should try to avoid this kind of, like, it will just try to take average around.
Just for a reminder, was CPD the one where where the
all the ranks are equal to each other?
Okay, yeah. All right, thank you.
Again, this is not the best one. It's the most simple one. It's commonly used, right? It's like, because people get used to it. It's already transferring from matrices to tensors took time, and transferring from CP to other methods will take more time. We will discuss in the next talk what are the limitations of CP. So now I will re-go this one, like missing data problems. So, and I want to give which kind of research, idea of which kind of research is available in tensor methods. One part is actually goes, there are different groups. One part very optimization based, one part is very algebraic geometry, because this stuff rank 1 tensor corresponds to second varieties and stuff, right? One part also very data oriented. So actually, whatever you are doing is you are generalizing concepts from matrices to higher dimensions. So if it's always good to start from matrices. For missing data, let's see what they are doing for matrices. Let's say I have a X, this is like data with missing entries. They do kind of, there are different ways for the matrix factorization based approaches. They do approximation, right? To low rank approximation plus a noise. But if you do low rank approximation, actually back in time, they were doing minimizing the rank of the approximation, right? It will already have imputed values, but this is not a rank, is not a convex, I'll say. Rank is not convex, so it doesn't give you convex problem. And people were trying different ways. One of the ones they tried, and it's very solid, is nuclear norm. So they said instead of minimizing the rank of the matrix, they minimize like some of singular values, and again, they minimize the nuclear norm of matrices. So lots of the groups, actually, they are trying to do this one for tensor level, right? I also work on this one, but nuclear norm of tensor is something you can approximate. There is lots of research here. We cannot exactly find it, right? Nuclear norm convex surrogate for rank, for matrices, for tensors as well, if you can compute. So I will just give, like, this is a question how, and I will give a case study which use this definition. They try to approximate. Nuclear norm for tensors, actually, let's say you have a three-way tensor, you are factorizing, and you are trying to minimize some of coefficients. This is the CP decomposition, lambda I times UI, VI, WI. Rank is equal in each domain, so we set I is from 1 to R. It's like sum of R rank 1 tensors, right? Every vector has a norm 1 here. Norm of UI, VI, WI is equal to 1. And we are trying to minimize sum of lambdas because they look similar to singular values of matrices. This is also another domain. What could be the singular value decomposition of tensors? Can you find orthogonal decompositions? This is actually also a different domain to work as well. So this is called nuclear decomposition, and people use approximations of this one because it promotes sparsity in tensor decomposition, encourages simpler low-rank representation of multidimensional data, and it's a big resource for data imputation. And for example, there is this study. I will go quickly because I want to speak about last cell interactions. They use for imputing single cell data. In single cell data, we have lots of missing points because of the dropout effect. They use different published data sets here, and they compare their method for Dr. impute, Saver, S-impute, Magic, CMF impute. This is consensus matrix factorization. Here, what happens actually, just to go quickly, they start from a single cell data and they get tensor out of it. Maybe just to go quickly over it. For one cell here, they are looking at Pearson correlation and they find K nearest cells. And they get a matrix for every cell. And they look at different type of, so these matrix, they vectorize every matrix. And they look at different kind of distances between these matrices. They find the P closest one. So for one cell, you have P matrix, K times M times P, right? And they do tensor imputation by approximating the nuclear norm. It becomes more powerful than the classical CP decompositions, because CP decomposition based imputation, they just look at the low rank and they integrate and they impute their data on these 5 different, actually there is more data set, and it's performing better than its kind of 2021 paper all that methods which were popular at that time. Okay, I have time or couple? Okay, so I want to quickly go over in the one of the last application, but there are more applications. Here, I like putting as the last one. So we will speak about self-signaling. So we know that communication is key. So I really like this game of the telephone. You start from some words and you whisper, then you whisper. Sometimes you can start like a message that I love mango smoothies that are super sour, and you can end up mangoes give superpowers. I knew it, right? So it can get complicated. So we don't, we want to understand actually how our cells are communicating. I hope they're not communicating like that. But sometimes when you have diseases or when you have cancer in the environment, maybe communication actually gets blinded, right? Communication gets problems. So it's really important to understand this ligand receptor interactions and communication pathway. So here, quickly, so here it is one of the type of the communication pattern we have, not the only interactions. And our sending cells send these moleculars, which we call ligand, and our target cells have receptors, right? Ligand cell and receptor should meet. And how we understand if these cells have ligand receptor pairs, we look at gene expression for corresponding ligand and receptor. And if these ligand and receptor meet, and it creates a response, right? And here I will see quickly case study 2022 paper, Tensor cell to cell. So this is to understand ligand receptor interactions for different contexts. So they have, again, their sender cell, receiver cell. They look at the ligand receptor pairs. Again, you can look at average gene expression for the specific ligand and receptor. They do also sometimes geometric means, different things to calculate communication score. But think about, you have different sender cells and receiver cells, and you are looking at different ligand receptor pairs. You have multiple matrices. They form a 3D communication tensor. And you can also look at different contexts, like time points, study object, body size, disease condition, and different contexts will give the fourth dimension. And actually, they are looking for 4D communication tensor. This context-aware communication tensor. Again, they applied Tucker decomposition in here. Again, I also have some questions about how to say improvement suggestions for that. And choice of the rank was not really given in there. And next talk, we will decide, okay, how to make sure about the rank stability of factorization. And they assume normal noise, which is not always true for genomics, but they were able to find ligand receptor pair activated in specific sender and receptor cells for different contexts. For example, what they were able to find. They identify multiple modules associated with distinct communication processes, like ligand receptor interactions linked to severity of coronavirus disease and autism spectrum disorder. And I just want to speak about something. Now we also have spatial data. It can be used in this context. So when we find ligand receptor pairs, we hypothetically say that, okay, I think this ligand receptor interaction must be happening. But in some cases, in this case, if these cells are not close to each other, it's not possible. So we have spatial transcriptomics data. Actually, you could also embed spatial maps, and you could also make spatially aware algorithm to find ligand receptor pairs. It would be improved version of this one. I give my research ideas. If you're working on it, invite me as well. Okay, last thing. So I couldn't cover everything. So I have a master class in University of Trento, November 25 to 29. It will be available over Zoom, which is free registration, right? So if it's like a 10 lectures, it will start from the basic, how we can apply these models. It will use different toolboxes. There are different toolboxes available for tensor methods. Tensorly is Python-based, TensorLab for MATLAB package, Tensor toolbox for, again, MATLAB. My group is using all three, so I will have to cover all three. So we will go through all these applications on real data sets. And our survey paper on tensor-based approaching for omics data analysis, application challenges and future directions coming soon. Amir is my PhD student, he's here. Daniel was in Broad Institute. Now he's in UPenn Medical School. Thank you. So as promised in the first talk, we are gonna discuss like limitations with some existing methods, and I'm gonna give a more probabilistic model, consensus ZipTF, which is like stable tensor factorization, which is developed for zero-inflated multidimensional data, which works perfect for omics data. we give like technical details, development of the algorithm and applications. Okay, so here we discuss some tensor methods. Now it's time to re-go over it quickly and then discuss some challenges with the existing beloved methods, right? So here we discuss like Kandekop parafact, just to remind, also some people might have missed the first talk, right? So let's say we have this tensor, like we are looking at how gene expression is changing across different cell types for different donors, right? It could be also across different tissues and different donors. It's like a multi-sample gene expression data. And like one of the simple methods we use, we said like Kandekop parafact decomposition. I will call it CP decomposition. We are reducing the ranks. You can start working with 20,000 genes. But again, what you care about like metagenes or gene cluster, gene expression patterns, maybe you have like 10 gene expression pattern, which is driven by different cell type conditions and donor conditions. That's what we try to find. Right, so we have, in this case, we will have number of donors times R. It will give us donor factors. And we discuss how we can use donor factors. Let's say I have 200 patients, but maybe there are 4 or 5 conditions, like for example, disease, non-disease, subtype of the cancer, which drives which drives different kind of gene expression pattern. So that's what we are trying to find. As donor factors, it will help us to find subtypes, like different donor conditions. And here actually there is there's a question
on Zoom.
Okay.
Yeah, maybe I'll ask it. Yeah.
Okay.
Yeah.
Okay, and then we will have gene factors and we have donor factors. Here, cell type factors. And we give kind of examples how rank 1 tensor looks like, but we will give more examples when we discuss application of our methods. So if people miss the first talk, they will still see like how to interpret this factorization. So again, what's happening mathematically? We have this tensor and we are writing this one sum of rank 1 tensor. So rank 1 tensor we set, actually we get this one like other product of these vectors. We have gene latent factor. If you have 20,000 genes, this one has length 20,000. Cell type latent factor, if we have 10 cell types, this one has length 10, and every entry is like the loading significance of that cell types for this profile. And like we have samples latent factor. Again, if you have 200 samples, then you have like 200 length vector here. So we have like, we take the other product of these ones, right? This becomes unit of an expression pattern, and we are trying to see how much of these expression patterns we have. We do low rank approximation. And so here there is, again, we actually approximate. Our tensor is an approximation plus a noise, right? We discussed this one actually. It is again getting 3 different matrices, one for genes. If I take all genes latent factor together, I will form gene latent factor. If I get all cell types latent factors together, I will get a big matrix of cell type latent factors. Does it move? Okay. I need to learn how to use this. It's like, and for the samples as well. Okay, what we didn't discuss next time. Here, classical CP decompositions and classical tensor factorization. Back in time, it's developed and it's heavily used in images, but it's okay to, in this case, assume Gaussian noise. But when we are working on like real world application, it's not like we don't always have a normal noise. So here, one of the questions we will address in this talk, how to truly incorporate the distribution of the data, which kind of noise assumptions we can have, right? Also, how to choose the rank. This is a really hard question. It's like, because I don't know to start with, I will have 8-10 expression patterns, right? How to choose the rank, how to test for you are really choosing like a good fitting, right? And which kind of constraints we apply. Some people apply orthogonality, but mostly I don't want to force gene spaces to be orthogonal to each other. I mostly assume non-negativity so that interpretation is easier. But to non-negativity, you should make sure that your data is non-negative. I know when you go through SCMPy, right, you're normalizing your data, suddenly you end up with negative numbers. So make sure it's like if you use non-negative factorization, you have non-negative entries. And uniqueness, again. So there is a great big debate in this area. So theoretically, tensor decompositions. But again, we are not really finding exact factorization. One thing actually, we are finding approximation. So theoretically, if you find exact decomposition, it's unique under milder condition. But we are in the real world, we work with noisy data, we find approximation. We will discuss the uniqueness in the next slide. Factorization will not be unique, and we will have to address it. Okay. So here, just to see what's happening in CP decomposition. So what we are trying to optimize. Again, there are different methods for the optimization. Lots of optimization groups are trying to solve in different way. I will give not necessarily best, but most common one. Most common, again, not necessarily best approach, most common approach. Sometimes common approaches are common because they're simple, right? So here for CP decomposition, I have a tensor, okay? I have a tensor and I'm trying to, I cannot use this one, approximate with another tensor, right? So it's like we are trying to minimize the error. We are trying to minimize the error, right? It's like we use the Forbinius norm, what we introduced in the first talk. It's like square of the entries and you're summing them and you are taking square root. And most of the papers, they are looking at explained variance. Like in PCA also, we look at explained variance to see if it is a good fitting algorithm, but we will discuss that this is not enough. We will go through different metrics as well. So they kind of normalize the distance here. Okay, normalize the distance and 1 minus distance. If the fit is 1, it's a perfect approximation. It means you recover everything. It's hard to find unless you are working on synthetic data, right? We always first test our algorithm in synthetic data. It's like, oh, we put everything and we recover them. So perfect, fit is 1 if you don't put noise, right? Because it should also account for the noise. And fit is 0 when the approximation is awful, nothing is, right? I mean, you wouldn't have 0, you would have 0 point something, right? Okay, but to start with, it's like I'm factorizing T to 3 latent factor matrices G, C, S with rank is R. This is not a convex problem. So it can be given as like 3 and minimizing. So this is like we are trying to minimize distance. It can be given as 3 convex problems. So to understand this for 3-way, if we have 10-way, it's 10 convex problems. So to understand this now, we will go back in the primer, we introduce unfoldings. For example, if I have a tensor of 10 times 20 times 6, I can write a matrix as 10 times 120. So I'm writing first mode unfolding. Then you can write 20 times 60. And this is second mode unfolding. Then you can write 6 times 200. Third right mode unfolding. So you get 3 matrices. Unfolding appears in the optimization steps. And so what the instability will come from? So we start with the initial guess. We have an initial guess for gene latent factor, sample latent factor, so latent factor for 3 modality. That's how you start for like alternating least square. Here I fix C and S. Okay, then I need to, so we are gonna solve for G, this algorithm, and we use like least square. Here, there is lots of tensor operations. I want to introduce this kind of operation as advertised in masterclass that I advertised in the first talk. Like if you're interested to learn more about operations, you can follow over Zoom. I think it might be also recorded at some point and just posted. So here we have Cartero products. It's like, look at these ones, these latent factor matrices, they have the same number of columns because the number of columns is equal to rank, right? If the columns are matching, you can take matching column-wise, we call it Kronecker product. Actually, you get the other product of vectors, right? Because I have R column vectors, I can just take the other products, C1, G1, C2, G2. That's how you get a bigger matrix. And here we fix for C and S, and we are solving for G. Then we are fixing for S and G, solving for C. It goes like that until you hit a threshold. So mostly it says, then it goes back and look at the fit or look at the distance. And it says, right, as in lots of algorithms, okay, if the result is not changing, passing a threshold, you stop. But this, since it's not convex, you don't have to go to global minimum as well, right? We will see in some stable cases, actually, sometimes you can get stuck. We call it swamp points. So it may also happen, and it's heavily dependent on the initial guess. And there is a big other area. I'm not very active in that area, but I want to be active. What is the good loss function? So when you say 2 matrices, let's say X and Y, how you can say distance between these two matrices. Actually saying distance in the same way saying similarity. How you can say this matrix X and matrix Y is similar to each other. Sometimes people look at the Euclidean norms like the Frobenius norm, it's the same, right? Squared errors, right? Mean squared errors. But in this case, you are only looking like in the Frobenius norm, are they numerically close to each other, right? But like we might also care, are they like as a distribution probabilistically close to each other, right? Actually, when you use the Frobenius norm-based CP decomposition, most of them you assume Gaussian distribution. Right here, there are different loss functions. People use Kullback-Leibniz. I'm not gonna go details, but if you go distance matrix with tensors, actually it's a big topic. Like sometimes they use more kernelized functions, right? And then they are looking at the similarity in this level. So it also brings some non-linearity. Just to say all the decomposition methods I explained today, they are multilinear because of also loss functions and structures. They can find multilinear patterns. But with these ones I discussed today, you cannot find non-linear patterns yet. Okay. So again, we will discuss these ones. And before going deeper discussion, and I want to say what are the classical approaches. Some people go through maximum likelihood approach, which is like, as we discussed, you are minimizing a distance and you put some constraints, right, in your factor matrices. You use your distance, like most common ones, again, is Frobenius and probabilistic one KL divergence, but there are also like different groups are trying more probabilistic distances. It's gonna be actually how in the next year's area will get shape. Okay, and there is Bayesian approach. Today we will use this kind of approaches. So here, most of the methods actually think that you have a normal distribution, but in the approach I will follow today in our method, I want to learn from the data. What's the distribution of the data? And I want to incorporate the true distribution of the data. So here, given the observed tensor, and we are gonna see tensor approximate ABC, is it likely, right? Like again, we are factorizing this one. And the goal in here is to estimate posterior distribution of the factor matrices, given the observed tensor T and any prior information you might have. For single cell data, we have loss of reference and we have loss of prior information about sparsity, right? And prior information we will specify for the factor matrices and for our tensor by kind of imposing some distributions. And we will have a hyperparameter set. We impose the distributions, but then we will actually ask the algorithm to learn about the true distribution. And posterior is this. Knowing your tensor and the hyperparameter set is the prior information. What's the probability when you factorize your tensor, you get these latent factor matrices, right? Again, not for saying anything, learn from the algorithm. Actually, this is like more probabilistic approach. The posterior distribution is analytically intractable. People estimate this one, they use techniques like Markov chain Monte Carlo, variational inference. We use variational inference. Maybe I'm not gonna go details. I'm gonna just mention about it. And with our method, in any case, we have the toolbox available. So we will discuss this once more. Okay, then I want to have a little bit more discussion on the numerical instability, right? Why even like we are trying to solve this problem. Like if there is no problem, there is nothing to solve, right? First, I want to convince you there is a problem, right? One thing to start is like, for the matrices, I said like lots of things from matrices, it doesn't generalize the tensors directly. For example, even the rank. Rank of a matrix, let's say 3 times 6, it has to be less than equal 3. Let's say I have a tensor 3 times 4 times 5, I cannot say rank is less than equal 3. But it's way more complicated. And the other thing is border rank issue doesn't appear with matrices. So if you have a rank 3 tensor, you cannot approximate, so rank 3 matrix, you cannot approximate this matrix by rank 3 matrices. There's nothing like that. So border rank is actually, let's say, for example, I have this tensor, this is 2 by 2. So actually first slide is 0, 1, 1, 0, 2 by 2 by 2. Second slide is 1, 0, 0, 0, right? So it's actually a nice cube. Okay, so this has rank 3. And you can write this one sum of 3 rank 1 tensors. This E1 is 1, 0, E2 is 0, 1, right? So it's kind of shown, this is the rank, you can find the exact exact decomposition, but it can be approximated by rank 2 tensors. So these are the rank 2 tensors. If you get the limit of this, it will give you the your tensor. So it doesn't appear in matrix. So what happens in this case when you sometimes run some numbers, because actually you are trying to find, like, you know that it's rank 10 tensor, but it approximates but rank 9 tensor, so all the numbers can explode. So it's like, don't worry, it's normal. It can happen. There is a numerical instability. It's not this tensor rank, they call not upper semi-continuous. There is a problem there. And one of them is the border rank. Other one we discussed convergence problem. After all, it's not a convex problem, right? It doesn't have to go the minimum, like that's a global minimum, and depends on the initial guess. So I want to show it here. Here I start from a rank 3 tensor, synthetically generated 3 times 4 times 5. So normally, right? So with a little bit noise, it should be very stable, right? Because I already know the rank. So even in this case, I run this tensor factorization with different initial guesses 40 times. And every time we run it, you can see in some cases, we can still get different fits. Different fits means you get different factors. So based on your initial guess, your results can change. But in this case, okay, so it's not like I reconstruct my images. It's like, you know, you work in biological domain. I want to trust my factors. I want to say, okay, I think I found a really important gene expression pattern. I need more funding. I need to go over it. I need to prove it. You should make sure about the significance of your factorization. So here, that's why I was more picky about the stabilization here.
So are you saying that when you try to pick a rank, you're not looking at reconstruction, but you're looking at interpretability of latent?
Yeah, so it's a good question. So we will discuss, I both look at reconstruction, then I'm also looking at the stability of factorization, both of them. Actually, we will define 4 metrics to look at to choose the rank. Okay, so kind of limitation, we said decomposition is not stable, especially for noisy data, but can you find non-noisy data? It's like convergence is not guaranteed. Rank selection is still a challenge. And it's like if you always say some Gaussian, right? You don't explicitly put the tree distribution of the data. And interpretation of latent factors also needs a pipeline. So here in the first talk, we also already discussed like tensor methods outperform classical matrix-based methods. Now we are going to discuss actually how to improve this classical tensor methods. Okay, so first thing I want to address is like a stability. So we kind of develop consensus-based tensor factorization. First, I will go through this, how to make sure the stability of the factorization. It will also help us to choose the correct rank. And later we will go through the Bayesian framework, how to incorporate the true distribution of the data. Right? Actually, this kind of models can be also applied. So actually, whatever you develop for tensors, you can also use it for matrices, right? Just, right? Even our toolbox, okay, it's okay for tensors of any dimensions, including the 2 dimensions. Okay, so here we have, let's say, I run this algorithm like 10CP decomposition, let's say 100 times, M times. Every time you run, you are going to get a gene latent factor matrices, number of genes times R, right? So you have a bunch of matrices. So I'm running multiple times now because I want to make sure the results I'm getting, are they like repeating? So if I have a very significant signal, that signal should keep coming back, right? For sure, if we are in a stable range. So for sure, it will not come as it is. It will come with some noise. Later we will cluster, but it should keep appearing. So here I will see if these different runs of the factorization consistent. So are they connected with each other? So first, for every factorization run, we are forming something called connectivity matrix. In connectivity matrix, for example, here we have R gene clusters. We are assigning genes to specific clusters. You can go in different ways based on maximum loadings is one of them. So we are saying if gene I and gene J is connected, if they are assigned to same cluster, right? They are gonna form a gene group. So in connectivity matrix is like IJ is 1 if gene I and gene J belong to the same cluster and 0 otherwise. And for every run, we keep repeating it. Every run, we are looking at connectivity between different genes. And at the end, we look at the consensus matrix, which is the average of connectivity matrices. So think about if everything is stable, let's say we get 2 genes, they should be always in the same cluster or always not in the same clusters. Numbers should be zeros or 1. Average should be mostly zeros and 1. And the moment you get dispersion between 0 and 1, then it means you have instability. So we are trying to see the dispersion. So it's calculated by something called cophonetic correlation. It's actually 1 minus dispersion. So one of our way to choose rank, for sure we will use explained variance still. We are gonna look at cophonetic correlation. So how connected different runs. And after that, it's not like we are going to choose the rank. We are choosing initial guess so that when you run the algorithm, it will not change a lot. So here it says actually when we are gonna concatenate all of them, it's like you normalize every latent factor so that norm is 1, so they are comparable with each other. You form a big matrix, but I claim that rank is R. So I claim that there are R gene clusters. It means they have to, right, cluster together, right? For example, as I said, yes, 1 gene latent factor will keep coming. It will come with the noise, but you can cluster it. So here we will find R clusters. Maybe I will also rediscuss one in the next slide. We will take the median. Maybe I can start from here. So we are in this point. Let's say A1 is our gene modality. We concatenate all of them, normalize. Here we are looking, are they clustering? First, you remove the outliers. It can be due to the randomness of the algorithm. And we are doing clustering. Actually, we went so simple k-means, but then different groups, they said, okay, your method was good. So you just went k-means, but they did more like more theoretical and nicer clustering methods. I can also give these papers as well. So here we are taking the median of each clusters, right? Because it's less susceptible to outlier, and we are forming our new latent factor matrix. The goal is I want to just count for the repeating signals. So after you form your latent factor matrix, like this is for genes, you fix your gene modality and you are solving for other 2, right? So you have a fixed initial guess for genes and you solve for the other 2. Like in here, right? It's like I get gene consensus, I fix it, and I solve for the samples and cell types. Okay, so again, since it also starts with an initial guess, for genes, there is actually, you go through different iteration, you can get slightly different answers, but they're not gonna be too different. But we are gonna measure it. It's not gonna be just saying, oh, it's not too different. We have experiments on it. Okay, we are gonna do some experiments on consensus if it's working or not. But so our algorithm is consensus zero-inflated Poisson tensor factorization. So now I wanna cover like zero-inflated Poisson part. Like consensus and added. Consensus approach you can add to any factorization method, right? You can factorize and you can add the consensus part. Okay, here for the Bayesian tensor factorization, these are tensor, this is cell types, latent factor, cell types, samples, and genes. So here, again, I mostly speak about Poisson and zero-inflated Poisson, but in our toolbox, you can change the probability assumptions. You can use different distribution. It's not just for Poisson. Okay, here what's happening. So we approximate our tensor. This is the assumption with rank R. We are saying it's like for gene expression is like the rate that you are capturing the genes. It mostly follows Poisson distribution, but people also use actually negative binomial, but it's very sparse. Sometimes like you have more zeros, excess zeros that you wouldn't expect from a Poisson distribution. So I want to also account for the sparsity. That's why we go zero-inflated, but we are having a gate parameter. We will adjust the sparsity. So here we say Tijk, like gene expression, like this is the entry, expression at gene I for cell type J for sample K follows the Poisson distribution, and the mean can be calculated by using this, rank R decomposition. And now, if you have some information about like latent factors, domain modalities, you can put it. We put gamma priors to induce sparsity. Sparsity also makes it easy to interpret. Also, it makes sense to induce sparsity for the gene factors. because not every gene is active in like pathways, right? So here we set a gamma prior for these latent factors, but you can adjust the shape and rate, and the algorithm will learn exact shape and rate of the gamma priors. This is for sparsity. We say our tensor follows zero-inflated Poisson. Mean comes from the factorization, and it might have, but it might have, you are allowed to use or not, more zeros than you expect from Poisson. Like it might have sparsity, then we have a gate parameter to adjust for sparsity. This gate parameter gives the excess zeros. So what are the benefits of these methods? First, we will discuss applications. And first, in the synthetic data, we will discuss, okay, did it work to, how to say, develop it? And it potential benefits to uncertainty quantification, incorporation of more realistic noise, and in a way, principled way to include prior information. Again, this is based on Poisson, but you can change the your distribution. Okay, so here I start from synthetic experiments, then I will go to more genomics data sets. And like I will discuss like our main projects that we use this algorithm. Okay, so first of all, to discuss, we have lots of like tensor-based methods as well. Does it have any advantage to go through like more zero-inflated Poisson-based distribution? So we call it zip distributions. So we have different examples in the paper. One of them, we created 10 times 20 times 300 tensor with rank 9. And for the latent factors, we assume gamma distribution with these shape and rate parameters, okay? And we are generating a tensor such that mean is given by this rank R factorization, and there is a varying probabilities of excess zeros. We change the sparsity level, right? We put different sparsity levels. And we are looking at explained variance. We are factorizing, finding a low rank approximation, and we are looking at how good is the explained variance. So we are using classical non-negative CPLS. There is also another method, sparse parafac. Parafac actually also stands for CP decomposition. It is developed for sparse data. They do L1 norm regularization to induce sparsity. We also tried this. There is like, for example, truncated Gaussian tensor factorization, again, assumes Gaussian distribution. This is the closest one. Gamma Poisson tensor factorization. It assumes Poisson for data, gamma for latent factors, but it doesn't count for excess number of zeros. Even that one failed. And our zip-TF zero-inflated Poisson tensor factorization. So when the probabilities of excess zeros are low, all of them performs well. But the moment probability of excess zeros increasing, since our method keeps adjusting the parameter, it stays stable. But other methods, their performance keeps going down. Here we compare with the tensor methods, then we will also in applications compare with the matrix methods as well. Here, because we like the idea is like why we are developing a new tensor method. Okay, so here it justified that we are going to use zip-TF, but do we have to use consensus? Now the next experiment will be zip-TF with consensus and without consensus. What we are trying to do with the consensus, when you run your algorithm, you want to make sure that in each run, you will get more or less similar results, right? So here we are running the algorithm 100 times. Here we are going from 0.6 for sparsity, right? Here, quite sparse data. We run zip-TF and see zip-TF 100 times. In every rounds, we are looking at the similarity between the latent factors. We look at cosine similarity. I mean, zip-TF is already like more or less stable for this algorithm. But we can see in C-zip-TF in the green one, similarities are increasing because you are really finding good initial guess for your gene clusters. And again, this is a little bit nuanced. Okay, the latent factors for different runs are similar, but are they similar to original signal? In this figure C, we are looking at similar to original signal. Consensus zip-TF actually gives very similar results to original signal. So zip-TF to count for distribution. Consensus approach, again, you can take this pipeline, put other methods as well to increase stability. Good. Now, this is the last synthetic data I have. Then we will go to lupus and Hodgkin lymphoma, the real data sets. So we use splatter simulation to generate synthetic single-cell RNA sequencing data set. This was like a quick check. Okay, does it work for genomics data? We have 1,000 genes, 3,000 cells, and 6 donors. And we embedded 5 gene expression programs defining cell type identities, cell type specific gene expression programs. And we also embedded 3 gene expression programs defining donor activities. So we have 8 programs. Then we will run our algorithm. We will see, can we really recover these 8 programs? So first thing for the rank, we are looking at explained variance and cophonetic correlation that we discussed during the consensus approach, right? So it kind of has, like in 8, we have good explained variance and cophonetic correlation, and we are looking, so we were clustering our latent factors, right? Through different runs. We were looking how good is this clustering. You can look at inertia, silhouette score to see how good is this clustering. We look, inertia has to decrease. For inertia and the silhouette score at 8 also, how to say, they agree with each other. So rank selection is 8, which is not surprising, to be honest, because we have 8 gene expression programs plus a noise. Okay, so we are gonna say, okay, rank is 8, but are we really recovering what is embedded as a gene expression program? Actually, I will tell, we're also doing with different log-fold changes for differential expression from low to high to see if the results are getting affected. I'm gonna discuss it. Here, for example, I have 8 latent factors, gene 1, gene 8. Let's say the G2, the right factor 2, is highly correlated with ground truth identity gene expression program 1. So we are looking at Pearson correlation. And the right factor 8 is highly correlated with ground truth activity gene expression program 1. One of them is identity, one of them is activity. Actually, so these are the right factors. This is the activity programs. All the factors correspond to one activity program. So it is able to recover actually gene expression programs. And again, we use splatter simulation to embed our activities and cell type programs. We use different differential expression log-fold changes, subtle 0.25 to very high 0.27, 0.75, right? But like we also have different levels to see, compare different methods. Here we compare with non-negative matrix factorization, amortized LDA, suggested for a reviewer because it's more deep learning based. Some methods appear because reviewer wants it, which is actually a very good method. This is non-negative CPLS. Consensus NMF, it's like NMF, they use a different way of consensus approach. And ZipTF is our model, CZipTF, our model with consensus. So actually you can see, it's like in both of them, we are looking at average Pearson correlation between our factors and the true factors. So it's actually outperforming our other models. In here, actually, we have more experiments. Good. Now, it's like I will discuss 2 applications. One of them will be lupus. And one application I will discuss in classical Hodgkin lymphoma to see actually biologically which kind of patterns we can find. Okay, so this one is public data set. Classical Hodgkin lymphoma is the project I start in Golub Lab. It will be our own data set. But for lupus, we are trying to unsupervised discovery of disease subgroups and multilinear gene expression programs in the peripheral blood of patients with SLE. Why I am interested? Because I have lupus. So I'm interested in lupus studies. So it's like we apply to CZipTF to multiplex single cell RNA sequencing data over 1.2 billion PBMCs from patients with SLE. So we have 3 groups. This is a really good one, right? We have 8 lupus patients with managed, right? Sometimes you have lupus, but you don't always have active flare. You feel like you're healthy until you get hit. And 8 lupus patients with active flare, and 8 patients like healthy controls. They don't have lupus, they're lucky. And we have 10 cell types, and we form our data, 24 donors, 5 cell types, and 13,525 genes. And notice that most of the time when you work on like gene expression data, people are reducing their genes to highly variable genes. Here we don't have to do that. Sometimes when you have also rare cells, you don't want to do that one as well. So we keep all our genes, like after some filtering. So here we look at the rank selection, you look at explained variance and cophonetic correlation, which agrees at 22. And we are looking how good is the clustering of the factors from different runs. Actually, we kind of look at inertia, silhouette score. So we kind of agree on 22. I will quickly see like which kind of important factors we get. Okay, so here, just to say, this is the UMAP of the cell types. So this one corresponds to one rank one tensor. So this column corresponds to samples, cell types, and this is like genes, but I cannot put all the gene factor. I'm looking at top 20 significant genes, and we run pathway analysis. For example, in here, we are finding T cell, like gene expression program. We run this one later for T cells. This is factor 10. Which is activated in almost all our patient profiles, right? We have healthy, black, SLE flare, like blue, and SLE manages pink. Again, we have another factor. This is like monocytes program activated in all our patients. So actually here, we are finding cell type identity gene expression programs with these factors. But we want to find something more. Can we find condition specific? Actually, we also have condition specific programs. Here, these are the conditions, like the UMAP of the conditions, healthy, SLE flare, and SLE manage. So we have some factors, for example, factor 21, one of them, it corresponds to patients with SLE flare, right? Factor 21 here, and you can see from the patient's profiles, it's really has higher weight in patients with SLE flare. And when you look at factor 21, it's also kind of, we do the pathway analysis. It's kind of, how to say, what we find as a pathway. I cannot move this one. It agrees with the literature. And factor 21, actually, it's like all the SLE patients. Too much doesn't matter if it's flare or not a flare. We also have SLE patients. This pathway actually gives type 1 interferon pathway observed in viral infection, which is commonly seen in lupus patients. So actually, it goes even more detail. Lupus patients with active flare has a different program. Manage has a different program. So this is important to know, because if we don't know the, again, labels of the data, we could, with this kind of methods, we can find it, right? But for differential express analysis, you cannot find it. You have to know the labels. For example, so we later run number of differential express genes was 55 when comparing SLE patients against healthy donors, and it's 122 when you compare patients with active flare to healthy donors. But if you don't know this information about the subgroups, your differential expression analysis wouldn't give a lot of information. So the takeaway from here, in scenarios where the source of intragroup heterogeneity is unknown, CZipDev can highlight subgroups based on expression profiles and identify gene expression programs driving the heterogeneity that may be missed by supervised differential gene expression analysis. And I still have time? Okay, so I will go through Hodgkin lymphoma. So this is gonna be, it's like our project with the Golub Lab. And yeah, because I was also postdoc at Golub Lab. So here actually, this factorization make us aware of labeling mistakes, because some programs were always, some patient factors, patient profiles were keep appearing in different gene expression programs. And we asked the clinicians, I will go over this example, is it a constant? But we figure out it's like labeling mistake. It can't even find labeling mistakes because it's totally unsupervised, right? It's trying to learn from data. Okay, so this paper first is like CZipDev with more applications. I worked with Vignesh, he's in Broad Institute in Golub Lab. Daniel was in Golub Lab, but now he's in UPenn Medical School. So you can see more applications and more technical details in the paper. And all the data, all the tutorials, everything is publicly available in GitHub. And again, you can also change your distribution and apply assuming different distributions. Good. So next application, this tumor microenvironment project, actually it's a very bigger project. We don't only use tensor factorization. I started discussing this project in 2020. I was thinking, okay, is it gonna take as long as like Boston big digging project? Remember they were reorienting tunnels, but hopefully it's finishing this month. I'm happy. Sometimes it can happen in Broad. You know, we have projects takes forever. It's like, so we are working on tumor microenvironments. Tumors are complex cellular ecosystem. You have diverse non-malignant cell types and states, like T cell, nature killer cells, you have also diverse malignant cell states. Here we have different cell types. We really care different cell states. We really also care about cell-cell interactions that we discussed in the first talk, like ligand receptor interactions as well. So here we work in classical Hodgkin lymphoma. This is a malignant B-cell lymphoma. So if you zoom in, you have these ugly-looking double nuclei cells. They're Hodgkin-Reed-Steinberg cells. They comprise only 1% of the tumor. So this is something unusual. Our bad guys, only 1%, 99% good guys, but they cannot go kill them. So they are surrounded by immune cells. Normally, immune cells are supposed to go and kill tumor cells, but actually in our case, immune cells are supporting the promotion of tumor cells. There's a reprogramming here. So they are difficult to grow in culture. They don't survive in immunodeficient mice. It means they need their microenvironment. So this kind of studies then works what's happening around this tissue. I'm not gonna go over it, but we also use lots of spatial data to see the organization. So, and the other thing, main therapy is very harsh chemotherapy. People survive Hodgkin lymphoma, but then they suffer, even if they survive, they suffer from the long-term effect of chemotherapy. Our goal was trying to find immunotherapy targets, which we were able to identify finally, before I became 60-year-old. So again, here we are using single nucleus data. We use so many different data sets. I'm gonna just presenting one piece. So it is 10 Hodgkin lymphoma patients, multiple replicates. Half of them has Epstein-Barr virus, half of them doesn't have Epstein-Barr virus. There is a correlation between having Epstein-Barr virus and having Hodgkin lymphoma. It's interesting because it looks very innocent virus. It's not that innocent, right? So here we also annotated 15 cell types, right? I will give more details about the data here. After some filtering, we end up with 19,000 genes, 15 cell types, and 40, when I say 40 donors, actually 40 samples, right? Because for some donors, actually, we are repeating different tissues, also different replicates. So I didn't want to also over-filter my genes because Hodgkin lymphomas are like rare cells in this group. I didn't want to, mistakenly get them out of the data. Okay, so here, the main goal, again, unsupervised stratification of these donors into subgroups and identification of gene expression programs that drive those stratifications. And in 1 or 2 weeks, this should be on bioRxiv with the name genome scale spatial mapping of the Hodgkin lymphoma microenvironment identifies factor required for tumor cell survival. I think name is very descriptive. I think we are keeping this name, but there might be changes. But it's gonna be like me and Vignesh, and it will come from the Golub lab. And for sure, there are so many people get involved from abroad. Okay, so here, again, I'm just gonna quickly go to results. Our metrics suggested start in rank 12. Okay, what we see in the rank 12. In the rank 12, in the lower ranks, actually, I want to give an idea what happens if you go lower or higher ranks. For example, let's say this is one, again, rank 1 Tensor. Okay, dark blue, Hodgkin lymphoma virus positive, open blue, virus negative, and you have reactive lymph nodes. Maybe I can quickly go over it. Actually, it gives this one. The last column, last row, I took it as it is. So this is the one we discussed in the first talk. Gene latent factors give JAK, STAT pathway promotes tumor cell proliferation and survival of tumor cells. Activated in tumor, again, height is based on the weights, and it is activated in, we are looking at the based on the weights, activating Hodgkin lymphoma patients. Okay, this is what happens in rank 12. And when you increase the rank, it's so beautiful. So I always suggest start from a rank and try to increase it. This signal divides into 2. It will divide into tumor for virus positive, tumor for virus negative. We increase the rank, and I will maybe rank 20, I will directly maybe go to result. And what happened when I increase the rank? There are again different factors, right? It's not gonna be just cell type identity. So it is gonna be condition specific. So let me maybe just speak about this one. So here we have a gene expression program. We actually biologically work on it and validate it for tumor cells, for mostly Hodgkin lymphoma with virus negative, except one donor appears in virus positive. And here we get another gene expression program, tumor cells for virus positive, one donor appears in virus negative, and we figure out this is a labeling mistake. That was the one. We're like, why this one keeps coming? Then we figured it's positive, it's negative. It was good. And actually, so there is also some heterogeneity, right? Like they join, but they have like different weights. And actually, we double confirm. Actually, if you use DG test, negative plus was positive, like significant genes you would get, it's concur with the genes we find with this factorization. And again, we find something. This is, I just want to go gene expression program for fibroblasts for Hodgkin lymphoma patients, and fibroblasts mostly for reactive lymph node patients, but there is some heterogeneity. And this heterogeneity can actually harm differential expression gene analysis, right? And another finding, again, monocyte program for Hodgkin lymphoma. Monocyte program mostly appears in reactive lymph node. Again, this unsupervised, an algorithm is able to find by itself. For sure, for this project, though, okay, last thing I want to do, people always ask what happens if you increase the rank a lot. If you increase the rank a lot, you're overfitting. You start finding a program activated in one specific donor you're not interested in. So you should find a sweet balance. So we start what the metrics suggests, but we kind of look at higher ranks as well. So in this paper, actually, I think a couple of minutes I can say, this is more general than tensor methods. We also use spatial data, cell types and states within the tumor microenvironment. Like what is surrounding these malignant cells. We use spatial data, which cells are enriched, which cell states and cell types, and what are the tumor-specific survival and growth signalings and mechanism of immune evasion, because we wanted to answer, get insights for drug-resistant metastasis immunotherapy response, and we are going to suggest some immunotherapy targets, work on intra-patient heterogeneity, crosstalks pathways within tumor microenvironment. We use actually tensor factorization for ligand receptor interaction. It was very powerful, and immunotherapy targets. And the last but not least, to give more ideas what's happening in this domain. Now, actually, deep learning approaches are very effective, but latent space is not interpretable. And tensor approaches are effective, but it's multilinear, nonlinear. Now, there is new communities. They're merging these two areas. Like they do tensor neural networks, or they use tensor factorization in these layers. So it is, I'm new in this area. I'm very excited. If you start working, invite me. Integration of tensor methods and deep learning approaches, it's a very good, promising area. For sure, if you're more theoretical, there is always need for theoretical improvements, both theoretically, like optimization, and you can find applications in many different domains. I know here people are most interested in omics data analysis, but like neuroscience, right? And like computer vision. Actually, we use a lot of images in broad as well. So there are lots of different applications. Thanks.